Hi everyone, we are talking about the citric acid cycle. The citric acid cycle is the central driver of cellular respiration. It takes acetyl-CoA produced by the oxidation of pyruvate and originally derived from glucose as its starting material, and in a series of redox reactions, harvests much of its bond energy in the form of NADH and FADH2 and ATP molecules. It is also referred to as Krebs cycle or simply TCE cycle. This cycle is the biochemical hub of the cell, oxidizing carbon fuels usually in the form of acetyl-CoA, as well as serving as a source of precursors for biosynthesis. So where as the body does this biochemical process occur? You may recall that glycolysis takes place in the cytoplasm. TCA, on the other hand, occur in the mitochondria, specifically in the matrix of the mitochondria. Remember that the mitochondria is the energy center of the body. Then let's take a look at the function of the TCA cycle. The main function of the citric acid cycle is the harvesting of high-energy electrons from carbon fuels. So how does this happen? It oxidizes two carbon units, producing two molecules of CO2, one molecule of ATP, and high-energy electrons in the form of NADH and FADH2. So let's see the link between glycolysis and the TCA cycle. Do you remember glycolysis? It is a process in which one glucose molecule is broken down to form two molecules of pyruvic acid, also we call pyruvate. Under aerobic conditions, so pyruvate is oxidatively decarboxylated by the pyruvate dehydrogenases complex to form acetyl. Well, acetyl is a coenzyme CoA, so we call it acetyl-CoA, which is the main substrate for TCA cycle. The pyruvate dehydrogenases complex links glycolysis to the citric acid cycle. So let's see the synthesis of acetyl-CoA from pyruvate. The conversion of pyruvate into acetyl-CoA consists of three steps. So step 1 is decarboxylation, and step 2 is oxidation, and step 3 is the transfer of the resultant acetyl group to coenzyme A, so short for CoA. So also a fourth step actually is required to regenerate the active enzyme. So let's talk about the decarboxylation. Let's look at the decarboxylation process in the conversion of pyruvate to acetyl-CoA. In this process, pyruvate combines with TPP and is then decarboxylated to yield hydroxyl TPP. This reaction, the rate-limiting step in acetyl-CoA synthesis, is catalyzed by the pyruvate dehydrogenase component, we short for E1, of the multi-enzyme complex. And then let's go to the oxidation. So at the oxidation phase, the hydroxyalpha group attached to TPP is oxidized to form an acetyl group while being spontaneously transferred to lipoamide. It's a derivative of lipoic acid that is linked to the side chain of a lysine residue by an amide linkage. This reaction also catalyzed by the pyruvate dehydrogenases component we call E1 and yields acetolipoamide. So let's go to the formation of acetyl-CoA. After oxidation, the acetyl group is transferred from acetyllipoamide to CoA to form acetyl-CoA. Reactions are catalyzed by dihydroxylipotransferase, so it's short for A2. Note that the thiamine-ester bond, which is energy-rich, is preserved as the acetyl group is transferred. And then let's go to the regeneration of oxidized lipoamide to CoA. This step is important because pyruvate dehydrogenase complex cannot complete another catalytic cycle until the dihydroxylipoamide is oxidized to lipoamide. In this step, the oxidized form of lipoamide is regenerated by dihydroxylipodehydrogenase, it's E3. 2 electrons are transferred to an FAD prostate group of an enzyme and then to NAD+. So note that this electron transfer from FAD to NAD+ is unusual because the common rule for FAD is to receive electrons from NADH. Because the common rule for FAD is to receive electrons from NADH. This unusual phenomenon is made possible by its chemical environment within the enzyme, enabling it to transfer electrons to NAD+. So let's go to the oxidation of carbon in the citric acid cycle. The conversion of pyruvate into acetyl-CoA paves the way for the initiation of TCA cycle. The TCA cycle begins with the condensation of a 4-carbon unit. It's called oxaloacetate, and a 2-carbon unit, the acetyl group of acetyl-CoA. This reaction, which is an aldol condensation followed by a hydroxylysis, is catalyzed by citrate synthesis. So the hydrolysis of the thioester is called Citric-CoA, powers the synthesis of a new molecule from 2 precursors. Then let's go to the regeneration of oxaloacetate. The regeneration of oxaloacetate from the reaction of 4 carbon compounds is the final stage of the TCA cycle, and it involves 3 steps. It's an oxidation, a hydration, and a second oxidation reaction. A methylene group, CH2, is converted into a carboxy group, CPO, in 3 steps. So oxaloacetate is thereby regenerated for another round of the cycle, and more energy is extracted in the form of FADH2 and NADH. So oxaloacetate is regenerated by the oxidation of succinate. Okay, so that's all for today, and we will see you next courses.